Hello, my name is Lucas Kogut. I'm an MD graduate from the Medical University of Lublin in the American program. And I'm going to give a little talk about COVID-19, also known as SARS coronavirus 2. Currently, right now, I am performing a clinical observership under direct supervision of Dr. Bauer at Harrison Health Center in Bremerton, Washington. And I've been tasked with developing these slides as this crisis continues to spread, and more people are being acutely aware of the scenario and situation going on out there, but maybe a little less aware of the exact inner workings and details and what certain misconceptions may be out there or questions. So I'm going to do my best to try and answer as many questions as I can through a series of hopefully 3 videos that will be condensed and not as time consuming as my previous video that I aired with all these slides before. So this is more of an accurate way to have everyone out there read this and follow along while I discuss the current prevalent issues. But what we're doing first and foremost, we're going to go over COVID from start to finish in an in-depth, basic, comprehensive dive or overview. And the way I've titled this is the ABCDs to COVID-19 or COVID-19. Just my kind of play on words there. So let's get started, shall we? Before we get started, I want to kind of just frame the slides the way that I have them formatted here. We're going to go over these points as such, expanding where we can on certain topics, and I've tried to include as much detail in these slides as possible. I am racing with the virus, so to speak, to give you the most updated information available. But with that being said, we have still limited information and data, even though this is spreading at a global pandemic rate. So I've done my best to keep it topical, and we'll hopefully keep this short. All right. So starting with today, we're just going to go over this main portion I brought up here, which is the definition, description, origin, including the brief history of it, and we'll discuss the snapshot of viral replication in the next sequential slides, along with pathogenesis and transmission. That'll be during the second video presentation. But with that said, I want to zero in on the first step before we dive into viral replication. That's discussing the overall morphology of what this virus is and the structural proteins that comprise it. And then in the sequential slides, we're also going to discuss a super spreader, what is this SNL type strains, and also discuss the very idea of strains, but more so debunk the SNL theorized strains that are talked about in the media and elsewhere. And then we're going to go over flu versus coronavirus, the Mythbusters edition, so to speak, or a side-by-side comparison to give everyone out there a more tuned-in differential and to help aid clinicians in a more deductive reasoning to see how these symptoms stack up, and also to everyone else out there who's wondering, okay, do I have the flu, or is there something more serious I need to be concerned about? So we'll talk about that symptomology side by side together. And again, those will be in the next video series that I'm going to post. And again, peds, OBGYN, so the obstetrics, gynecology, and pediatric populations, statistics thus far known, and special precautions and scenarios to look out for as per the updated guidelines for CDC and WHO organizations. And we're going to also briefly go over the healthcare personnel post-exposure guidelines and PPE, personal protective equipment guidelines, and what's going on there. I'm not going to dive too deep into that because all that information can be readily found on the CDC website under the healthcare personnel subtext. So I have a link included, but we're just going to go over the general big picture of it, and discuss that at length further in this lecture series or presentation series. And then the big topic of testing, and this seems to be an area of ongoing debate and concern, and we're going to try to address the big issues that are surrounding the testing and hopefully address all the points as accurate as I can. And then we're going to go over treatment and precautions and the overall summary of what this whole presentation was about. And maybe a little bit of a fun bonus round fact versus fiction questionnaire. So, what is COVID SARS coronavirus 2? This is where we're going to frame in the description, definition, history, snapshot of viral replication, pathogenesis, the transmission. Now, as I said, today we're just going to go over the description, definition, with the origin story and the history because that takes a little bit of time, but I find it very important to go through that information so people are more aware of how this came to be and how it's affecting us today. And it's hopefully going to answer a lot of questions that some of you may have. So, coronavirus, by the order of Nidovirales, which also comprises another viral families, of which I did not include the name because it's not important and relevant to today's discussion. But also, furthermore, though it belongs to order of Nidovirales, the family is Coronaviridae, of which the subfamily is Orthocoronavirinae, and there are even further divisions of 4 groups denominated by the alpha, beta, gamma, and delta, and we're going to discuss that again in the subsequent slides. So, coronavirus infectious disease 2019, also referred to as severe acute respiratory syndrome coronavirus 2, is a new variant mutation of the coronaviruses that have been identified in the early 1930s from poultry populations, and that particular strain was infectious bronchitis coronavirus, IBV. And again, all these viruses, as we'll soon find out as we go through these lectures, are primarily found in animal hosts. So a lot of the information we have pertaining to coronaviruses today are from the animal populations and not so much humans. We have a small pool of viruses known to infect humans that are of the coronavirus family, and we're going to go over those and discuss those a little bit further. But I just wanted to give a background origin of where these have been found from, and that's from the animal species. And so, again, poultry populations in the U.S. in the 1930s, and then other animal species have been discovered to be host to the coronavirus strains in the 1960s, roughly. And it's a single-stranded positive sense enveloped, encapsulated, basically that's what that means, virus that has been given its name due to its crown-like or halo-like appearance of its glycoprotein capsid, which has these numerous little spike-like projections that I'm sure everybody out there has seen by now of that beautifully rendered picture, or unbeautiful, however you want to term it. But we're going to go over the structure and see that picture again in more detail in a few slides to come. So, and also we're going to talk about the genomic structure as well, and talk about what a single-stranded positive sense RNA virus is, and how that compares and differs from negative-stranded RNA viruses. It's also important to note, I did not write it in these slides, but it's non-segmented, and we'll discuss that as well coming up. Okay, and COVID-19 is the most recent virus causing widespread infection in humans to date. Duh. Everybody knows this, right? Everybody that's out there knows this. So what's more? What's more to know? We just talked about it being in the animal species, primarily on these coronaviruses, and so it can be termed zoonotic in origin. So zoonotic meaning that it is an infection predominantly in animals and stays within the animal species and not so much to humans. And when we pick it up from animals, that's when it becomes particularly pathogenic, when we are not the intended host for its normal life cycle. And a good illustration of that is that it somehow is found to live its normal life cycle within bats, as we'll discuss coming up again in the next few slides. So next what we're going to talk about is this story, which is a origin story, so to speak, about the history of coronaviruses, what we know about them so far, and I feel it's important to dig a bit deeper and use the information we have through an article or submission published on the NLM NIH PubMed website that is called up here at the top left corner, Animal Coronaviruses Lessons for SARS. And the way that I like to go about talking about these viruses is always starting from the beginning, because more and more details can often be found when we dig enough in the past about what we know so far. And that has a lot of influence on the treatment modalities and effective management strategies that we can deploy going forward to address the current COVID-19 outbreak. And so I know that there's already a lot of information out there, and if you've already known this information that I'm going to share, I apologize, but just a bit more about why I'm taking the time to post these slides and lectures for everyone out there to listen to and maybe derive some new information from this, because I have a genuine passion and care for helping promote common well-being and overall health for every human out there in the globe, and that's why I got into medicine. And so the best way that I find that I can contribute is by being able to maybe shine a light on some of this confusing stuff that some people maybe only having seen media advertisements and information that seems a bit murky or scary, or maybe it's being downplayed. So my goal is to strike a perfect balance of where we need to be in addressing COVID-19. Should we be overreacting quite a bit, or should we be just not worrying about this much, going on, carrying on with our day? I want to have us somewhere in between, in the middle of being or treating this like a serious issue, which it is. And hopefully by the end of all these slides and my presentation, that is what I can contribute the most to, is promoting a overall sense of taking this as a serious threat and helping combat this through education. So with that being said, let's get started, and I hope I can maybe teach you something a bit new, maybe you didn't hear about before from any source out there. I know I'm racing against time with this, as more and more information is thrown at us each day. So if I haven't shown you anything new, I apologize, but I do thank you for taking the time to listen to this. And I understand, if you're a clinician out there or a healthcare worker listening to this, I greatly appreciate your time, because as I very much know and understand, you may not have a whole lot of time in your hands, especially now trying to address the public and treat as many patients as you can. So again, thank you so much for listening to me, and I hope that I can make use of your vital time efficiently. So let's move on here. And so what I've written down is, again, we understand very little about these viruses, and we furthermore have only a few of these strains, these coronavirus strains, fully sequenced for phylogenetic analysis to trace their evolutionary origin. And phylogenetic analysis just means a family tree. It's one and the same. So tracing it back to the grandfather strains. So what I go on to explain is, what we do know is that cross-species transmission amongst diverse animal types occurs quite often with coronaviruses. And this is important to understand, because though I didn't include it in these slides, there was evidence of a beluga whale that was autopsied and found to have coronavirus virion particles in its liver tissue. So that just gives us an idea of how far-reaching these viruses can be, affecting all types of animal hosts, and humans were no different. So let's move on, talking a bit more about this. So I've kind of used the terminology grandparent strain to illustrate this a bit better, because these are truly kind of the grandparent strains we've discovered in the past, and again, mostly in animal populations. But it's interesting to note, and I've started off here with the PEDV, porcine epidemic diarrhea coronavirus. It has a close genetic relationship to human coronavirus 229E, which we're going to discuss at length in the next slide. But it's one of the 4 human coronaviruses that are implicated in causing the common cold and during the late winter spring seasons. And we're going to talk about that a bit further. But the other strain, grandparent strain, so to speak, is the porcine respiratory coronavirus. And for those of you who don't know, porcine is referring to pig populations. So pigs as in, the swine or sows or oink oink. You get the picture. I'm not sure if my pronunciation is clear enough through this microphone, so I just want to be clear on that. Pigs as in pigs for bacon. Though I don't know if you'd want to have bacon from these infected pigs. Although I don't want to misrepresent any kind of information there. There's been no evidence that it's transmitted through food as of yet, unless someone that you know is ill and actively coughing on your food. There may be some risk there, but as far as we know, it's respiratory droplet transmission. So again, don't want to worry anyone about having your bacon. You can go ahead and enjoy that all you want. So now there's the PEDV and PRCV. As we see in their names, the PEDV has diarrhea in it, meaning it predominantly causes gastrointestinal type symptomology, diarrhea, nausea, etc. Whereas the PRCV causes a predominantly respiratory symptomology because it affects the respiratory tract. Now, interestingly, this does not mean that they cannot that they couldn't go on to infect other tissue types. For instance, PEDV could certainly cause infection in the lung tissue, and vice versa. The PRCV could infect the gastrointestinal tract. That's because these viruses, as a part of their genomic makeup, have acquired such tissue tropism, and the tropism means the affinity or ability to infect other tissue types. But for right now, and for simplicity's sake, PRCV, respiratory, PEDV, diarrheal, or gastrointestinal, small intestine. Now, moving on, we come to the more sinister grandfather type. This is the grandfather that is just really bad, kind of like The Godfather. Even though Godfathers were great movies, this is the bad guy, so to speak, of these strains, and is known as TGEV, or transmissible gastroenteritis coronavirus, which is a more virulent form of the coronavirus grandparent strains, of which PEDV and PRCV are said to be an S gene mutation of, or S spike protein. And right now I want everyone to key this into their minds for the time being. When we refer to S gene mutations, S spike proteins, or ancestral even, we're generally just referring to the mutations that affect the spike protein, because that, as we'll see later on, directly affects their ability to infect humans by binding to host cells and subsequently entering the host cells. So when we talk about these S genes, S spikes, it's the little spiky projections that everyone's seen out there on your TV. Okay? So when we go on to discuss that the transmissible gastroenteritis coronavirus strain causes primarily, the gastrointestinal symptomology through its infection of the small intestine epithelial cells, causing severe villus atrophy. Now, atrophy meaning when you lose it, right? In a general sense. It would be the same thing as someone who's laying on a hospital bed for months and months and months, or who's in a coma. Their muscles will atrophy over time, or it's if you don't work out for a while, or you have a very stationary lifestyle, you can't move, your muscles will then start to atrophy and wither away. And that's what's happening in the gastrointestinal tract here at the small intestinal epithelial cell level. They undergo atrophy, though, at a much more rapid pace, and this results in malabsorptive diarrhea and a potentially fatal gastroenteritis, because if you can't absorb your essential nutrients, your fats, your electrolytes, or to be able to maintain fluid balance, most importantly, you're going to bottom out your blood pressure and through all the other electrolyte abnormalities succumb to the infection quite rapidly. So it's a very, very virulent strain. It's also been shown, however, though, that TGEV can involve the upper, and I put this all in caps because it's important to square this away in our minds. So it involves the upper respiratory tract, leading to transient shedding of the RNA in the nasopharyngeal passageways. This is important because, as we currently know, compared to COVID-19, which is a far down the line derivative descendant of these viruses, affects primarily the lower respiratory tract, which we'll talk about at length coming up. It's important to understand that when we go to do these nasopharyngeal swabs, which is how we test for the flu as well, because the virus sheds in our nasopharyngeal passageways and also our oral mucosa through sputum. So, say that COVID-19 has a mutation that's more closely related to TGEV and, or rather, is not closely related to TGEV, and you are shedding it in your upper nasal pharyngeal passageways, you may miss the testing for the virus. So we're going to talk about testing all that at length further to come in the next video, but just to give you some foreshadowing, this plays back into the idea of the tissue tropism, and it also lays down the framework of viral shedding, and we'll talk about when that peaks and all that further to come. So, and I also put here, it curiously spares the lower respiratory tract from significant disease, the TGEV that is, and that's important for comparison with current COVID-19. So I go on to say, of interest and current concern is the fact that these viruses are more prone to mutations that benefit their ongoing pathogenicity, crucial to their survival and thus persistent infectious process within their host. And if that sounds like a mouthful, I understand. It kind of is. I'm not been the best at my run-on sentences, but there's a lot of important information in that sentence. And I'm just going to go on and continue here of what I've written down, is meaning the longer they are present within said host. And I use all caps for longer because this is going to be an important discussion right now. As we're going to see again and again in the slides to come in the next videos, what this is saying is that these viruses, through evolutionary changes and their high rate of mutation and recombination, and the mutations that benefit their ability to undergo efficient recombination, is what allows them to be able to persist in their host for long periods of time. And the best way they achieve that is they pick up mutations that have benefited their ability to block their discovery by their host immune system, both the innate and the acquired immune systems. The innate immune system, for those that don't know, is our general first-line defense immune system. It involves a certain cell type squad, if so to be frank. That would include the macrophages, our dendritic cells, which dendritic cells are just sort of specialized macrophages located all throughout our body that are designed to pick up infections wherever they may be. And also they involve some of the natural killer cells, and so that without going much into the immunology, that's basically all I need to know for the innate immune system. There's a lot more to it, but for general sense, that's all I need to know for now, with pertaining to our discussion currently. And the acquired immune system is obviously our memory cells, our memory immune system, the ability, so these cells that take in this virus and start developing a code to produce a right antibody to target and neutralize it. And that takes some time. That takes some time to be able to recognize that sequence, especially when this virus has never been in our bodies. It takes time to develop that. And furthermore, it'll be even more difficult to develop an acquired immune response when this virus has ability to inhibit our acquired immune response. So, and we'll talk about how that happens when we go through the replication, snapshot, and subsequent pathogenicity lectures or presentation. So, again, it's a survival mechanism these viruses use very well. And I'm going to repeat the sentence here that says the key point to highlight as to why this occurs so often amongst these viruses lies intrinsically to their ability to utilize the beneficial mutations for altered host tissue tropism. And this is through interaction with other viruses of its own family, so coronaviruses out there, or also from other RNA viruses known as the RNA quasi-species. So this could be from influenza or all the other RNA viruses that can be present in a host at one given point in time. Though co-infection is quite infrequent, and we'll talk about that later on as well. It's about a 2% co-infection rate, meaning to have influenza and coronavirus at the same time, although that may be changing as more information is discovered. Anyways, that essentially wraps up all the information we need to know about these viruses as far as their origin goes from their grandparent strains and how they've been able to adapt and evolve over time. And moving on, we're still stuck on the history origin story, so I know, I understand this is turning into one of those History Channel specials, but just stick with me and hopefully this will become more clear and you get a rounder sense or a more well-rounded sense of these viruses and where they came from. It's quite interesting really. So, historically there are 7 coronavirus subtypes documented that have been linked to illnesses manifested in humans. Okay, so 7 known to humans, of which 4 of these, Magnificent 7 or Unmagnificent 7, sorry Chris Pratt, a movie for you here, they've been known to cause the common seasonal cold or symptomology of colds out there. And these include subtypes 229E, which we talked about earlier, being in close relationship to the grandparent strain PDEV or PEDV, porcine epidemic diarrheal coronavirus. So, and these collectively account for roughly a quarter or 20% of the common cold cases exhibited or seen each late winter, early spring season annually. The remaining 3 coronaviruses, or what we can reference to as the quote-unquote heavy hitters, or what I like to call the unholy trinity, because they truly are unholy the way they cause infection. And as most recall, the first one on the line here is SARS coronavirus, which was responsible for the 2002-2003 Hong Kong outbreak, and is the closest related virus to the current COVID-19 virus, also known as SARS coronavirus 2 that we have listed down here. And it is the star topic of our show today. So, and in between these two, we've got MERS coronavirus, which is the Middle East Respiratory Syndrome, identified in 2012. And I'd like to just take a brief diversion here to talk about something called the case fatality rate. So CFR. Now, for those of you who don't know, or I'm sure everyone by this point understands from seeing this at one point or another, case fatality rates are just taking the number of dead or fatalities, however you want to reference that, over the total population that has been found to be infected or tested positive for the virus. So we'll term that as I. And this will give you the percent expressed of the fatality rate. So that's the way we discover that. And it's kind of important to note that the most recent of these three strains, aside from COVID-19, was the MERS coronavirus, which had a very, very high mortality rate, though it was relatively small of an infected population, and thankfully a limited spread due to the geographical location of it. But it came out to something around 34% of a fatality rate, which is very, very high, especially if you extrapolate that to a larger population. So thankfully that was able to be contained, and we haven't seen many cases develop since then. So fingers crossed, right? So, but a bit more on that is, and we're going to talk about this in the next slides, but it's good to foreshadow things here. So all these three have their main host has been identified to be bats. Okay, so we'll label that as B, and then they have to go to an intermediate host before they cross species into humans. So the intermediate host for MERS was a camel, so we'll label that as CAM, okay, for camel, and then subsequently goes into humans. That was the identified intermediate host and affecting humans here. So again, with COVID 2019, what has been a recent discovery, and we will see coming up here soon what the intermediate host looks like. It's from bats again, and then it goes to the intermediate host, which has been identified or theorized currently to be a pangolin, and I'll show you what that looks like in a bit. So label that P, and into humans. Okay, so all right. Now, after having put this all down here, even though intermediate hosts have been identified and then the transmission to humans after that, it's been theorized that since the original strain mutation that's from bats can cause direct pathogenicity and infection right off the get-go, it's been said that the need for an intermediate host may have not have even been necessary, though it certainly does enhance the theory is it does enhance the ability of the virus to jump from the intermediate host to humans due to another mutation it's picked up. But since the mutations we've found in the bat strains could be directly virulent to humans, again, it's not known whether or not an intermediate host is really all that necessary. So, and that's what we're going to talk about here again. So, this is your cute little guy, the pangolin, also known as a scaly anteater. And what I'm circling here is they're characterized by these peculiarly large protective keratin scales. And the reason it's theorized that this is the intermediate host, or suspected to be intermediate host, is because these, for one, it's been identified to have a SARS coronavirus strain within its body, and furthermore, they're found widely throughout all the wet markets for trade and being trafficked due to the fact that their scales are believed to hold medicinal purposes. So that is the reason why they are likely to be the intermediate host, widespread use for their scales. And again, this information here just goes over what I just discussed in the previous slide, talking about how was an intermediate host even necessary, and especially given the fact that it's been found that this virus likely made the transition from either bats or this intermediate host back in November 2019, and it could have been directly from bats to humans. So, as more information comes out, or maybe it's already out there and I've missed it, and if I've misspoken, I apologize. I'm trying to be as relevant as I can and as accurate as I can, but when you're dealing with such a rapidly changing situation, it's hard to fit in all the key details that may be developing second to second. So if I've misspoken, I apologize. But anyways, let's move on. So, moving on, we're talking about now, not moving on, I guess. We're going back to the slide before last and more. So we're just expanding a bit on the information we've already discussed and gone over, and adding a bit, more in-depth detail to that. So we talked about briefly the alpha, beta, gamma, and delta denominations, right? So how does that apply, and why is that important moving forward? Well, frankly speaking, unless you're a seasoned virologist, and I apologize to anyone in the audience who may be listening that is a virologist or a molecular geneticist, I apologize to you, and I'd like to take this time right now to say and state that I am not a virologist myself or a molecular geneticist. I just have a strong interest in promoting awareness and education about these viruses, and so, virology itself interests me, and the best way to put that interest to use is to disseminate this knowledge to everyone out there listening so we can understand a bit more what we're up against here. So, what I go on to describe is that so COVID-19 can be classified as a beta coronavirus, which also is shared by the other two bad boy strains, the human coronaviruses, which is SARS coronavirus 2002-2003 outbreak, and MERS. So all three of these are said to be a part of the beta coronavirus origin group 2b. And what's more importantly, and I can go into bold and underline, is this important information here, is that COVID-19 has been found to share an 89% nucleotide sequence identity to SARS coronavirus ZXC21, which is belonging to our little friends, the bats. So as we can see, bats are of high research importance to understand not only where these viruses came from, but how the bats have learned to live and adapt to be completely asymptomatic and living in symbiotically with these viruses. So that's of heavy research interest. What mutation in the bats has allowed them to live asymptomatically with them, or vice versa, what mutation the virus is exhibiting within the bats that is not particularly pathogenic to the bats. So again, it's something that we have yet to discover. And furthermore, COVID-19 shares a 50% nucleotide identity match to MERS coronavirus, and finally an 80 to 82% nucleotide identity to SARS coronavirus. And that's why we give its name SARS coronavirus 2, for simplicity's sake, also known as COVID-19. And what's important to take away from all this is, as we see, just by the relationship, this further reinforces the fact that we're going to try and drive home each slide as we move along here, is these viruses are very, very good at adapting to different hosts and still maintaining traces of their origins in their genome to one another. So that's of high importance, again, to stress the issue of isolating ourselves to keep this virus at bay and stopping it from mutating and mutating and mutating to something that can be elevated from the serious that it is right now to something that is critically serious. If you're already not considering it critical, which it is critical, but it could get worse, all right? That's what I'm trying to drive home here. here is that it could get worse. So isolation is strongly stressed. So I apologize if I continue to reference the isolation thing, but my hopes is maybe it'll be a type of subliminal messaging to anyone listening out there. So just be aware of that. And moving on, I talk about what, so far what we're going to discuss is, over the next slides, is the general replicative strategy that these viruses exhibit, and to which COVID-19 also exhibits largely, just the overall similar replication that they all experience and go through. There are some differences, and we'll discuss those later on when we get to the replication. But again, the discussion of the replicative process will be discussed in the next presentation of slides. And what I'm kind of foreshadowing here at the bottom is I'm going to say that we're taking a little detour before we get into the replication to discuss more of the morphology, and that'll be discussed at length in the next slide. And it's important to have a general overview of the structural proteins before we dive into the replicative process, so we can pull as much information from what certain pathogenic factors these viruses have just on the outside of their viral appearance. So, are we ready? Let's get started. And so here we have it. This is even though it's titled Snapshot of Replication, again, that's going to be the next video on the replicative process and pathogenesis, but right now we're discussing the morphology. Sorry for my penmanship, but I got it there. So, before we dive into the deets, we got to understand as much as we can about this little ugly guy, right? So, and I'm sure everyone out there has seen the picture, and I'm going to show it to you again, but we're going to hopefully go over it together in more detail, pointing out to you what each of those blobs on it look like and what they are, okay? So, and I talked about this earlier on, about that it's a positive sense single-stranded RNA, although I didn't include in here is also the non-segmented determination or denomination of the virus. And so, just to explain what a positive sense single-stranded non-segmented RNA virus is that it's essentially a messenger RNA, which host cells, normal us, our bodies, humans, animals, we have messenger RNAs that are translated directly into cell proteins and enzymes. And so this virus is essentially already a preformed mRNA ready to infect a cell and directly give rise to its infectious proteins and enzymes and just continue the pathogenic process from there. So it gives it the ability to create infection quite rapidly. So that's why you can see sometimes the incubation time being very small, or a few days or longer. And to dive into that quickly, here's the variation in the incubation time. The longer it has an incubation time, goes back to what we talked about earlier. Their ability to persist in host cells is what allows them to survive, right? So the longer that they can sort of remain in a host without detection, making the host asymptomatic, is beneficial to them. So they are still replicating and creating their proteins and infectious processes, but doing it silently by the ways that they inhibit the immune system. And we're going to talk about that at length during the replication discussion. All right. So just for now, let's dive a bit deeper into or talk about the morphology here, starting with completing the RNA structure. So what's interesting and unique with these viruses is they have a very large, on the order of some 30,000 kilobase nucleotides as an RNA sequence, which when you're talking about RNA viruses, that's very large. And it's been referenced that, the genome varies from about 28,000 kilobase pairs to 32,000, but for simplicity's sake, just remember the number 30,000, which is still very large. And again, we know the appearance. It's not Corona extra beers. It's Corona extensions. So I'm just trying to catch those of you who may be falling asleep out there. We're not talking about the beer. We're talking about the little spike-like projections coming from its nucleocapsid, or the glycoprotein capsid. And I go on to discuss here that, we know this, right? So what gives? What else, man? What else can you tell me? Well, I'll tell you. There are some important structural proteins that coronaviruses exhibit, and the four main ones are as such. We have the S, or spike protein, and we have the M for the membrane protein, E, and N. And to go on to talk a little bit more about each one of these, the spike protein, all those little projections we see from them, are what allow the virus to, again, attach and gain entry to the host cells and create infection. And the M protein, as we'll go on to show you in the image coming up, they're little orange blobs closely associated to one another, and they help create the overall membrane integrity and structure of the virus itself. And the E protein also functions in making up the integrity and structure of the virus, but it also has some very important things to key into, and we're going to talk about those in just a second. And it's a very small protein, and it will be the yellow in colored little tiny blob on the picture coming up. And as for the N protein, which is the nucleocapsid protein, this is the protein that houses the very large RNA genome. And I don't know if any of you have seen my previous video. I have my own artist rendition of it, so I please advise anyone who did see that picture or drawing, don't use that as a reference. That's just the way I picture it. It's by no means the actual appearance of it. And what I've written here next is that some, but not all, coronaviruses also have a fifth structural protein. And new evidence has been suggesting, and as we'll see in the next picture, it looks like it's also present on the COVID-19. But largely right now there's still debate as to whether or not it does in fact produce or possess this fifth structural protein known as the HE, hemagglutinin esterase protein, which the best way I can illustrate this is like this virus has a little squirt bottle of acid right next to their little spike-like projection, and all it does is secrete this acid, or the hemagglutinin esterase protein, directly onto the host cell membrane, eats away at it a little bit, and makes it much easier for the S spike protein to come in now and bind and enter the cell much easier. So, and I go on to write here, it has been shown to aid in mucosal cell entry via enhancement of the spike protein. So, and as I put here in parentheses, it's not known yet whether or not COVID does possess this fifth structural protein, but as more and more evidence is coming out, it sounds as though it may in fact have HE on its cell surface, or virus surface. And coronaviruses, coming back to their very large genome, it's been, we can subdivide it into two main divisions, and approximately 2/3 of its genetic makeup or genome, roughly 20,000 kilobases of the nucleotides, encodes for the structural proteins, what we just talked about, the S, E, M, and N proteins. And the remaining 1/3 of the genome, roughly 10 kilobases, encodes for the non-structural proteins and accessory proteins, NSPs and accessory proteins. Many of which are still under research for exact pathogenicity, and they're in the articles that I've included up here. You can go and read if you care to. There are certain NSPs that are numbered 1 through 16 or higher, and suspected of how they affect certain processes, but still more research is needed to find exactly how they create infection or do what they do within the host cell. So we're largely going to leave the numerical categorization of these NSPs out of the discussion for simplicity's sake and for not tiring out our neurons, because it's very extensive and it doesn't really give us much benefit as far as what we're trying to accomplish in this discussion today. So with exceptions to one non-structural protein that I will include numbering, and that is number 12, and we'll talk about that in the replication talk. And also, what's unique with these viruses is they have a helically symmetrical nucleocapsids, which is unusual or uncommon for positive sense single-stranded RNA, and more so a characteristic of negative sense single-stranded RNA. Further driving home the point that these are the experts as far as viruses go in their ability to undergo recombination and mutations to pick up all these other virulence factors from all the other RNA viruses out there. And negative sense viruses are subject to giving their virulence pathogenic factors to the virus, and that's how we see it here. It's helically symmetrical nucleocapsids, which is uncommon and more common for the negative sense RNA viruses. And so in the next slides, we're gonna take a deeper dive into the structural proteins and reference them again and again as we move through the replicative process. But right now, I want to do a bit of foreshadowing and discuss at a little bit more detail the E protein, because the E protein has some clinically significant value here in both the testing realm and pathogenicity realm, and it's going to come up again and again throughout these slides. So the E protein, or the envelope protein of the virus, has ion-like function, meaning it can function like an ion cell of most cells in our bodies. And with that ability, it can alter the host cell's secretory pathways, aiding in its ability to be subsequently released after it's packaged up in the viral progeny known as VLPs, the virus-like particles, or virions, however you want to term those. So it has significant importance when it comes to the spread of the virus throughout the host it's infecting. And I further go on to highlight and bold that here is what's important is the recombinant forms that lack the E protein are not always lethal. And that's important in the sense of a reassuring factor, but also a bit ambiguous in the sense that even though certain virus particles from one portion of the host tissues lacks the E protein on the surface, it may still have the E protein incorporated in other cells it's infecting somewhere else within the body. And this has relevance when we're talking about the testing modalities, because certain tests only test for certain, a couple parameters. And for one of the tests that we're using today, and although at the time I'm posting these slides to everyone out there to watch and have available, that may be changing, but for one of the tests that uses PCR, it screens for two parameters, or two proteins. The E protein is one of them, and the RDRP, the RNA-dependent RNA polymerase gene, which aids in the transcriptive process we'll talk about later on. Now, the test before has been said if it's negative for one of those proteins, it was classified to be a negative result. And as we see here, that's not so good, right? Because as I go on to describe, though it's not required for viral replication, it is required for pathogenicity, meaning one of two things here. The virus can still replicate, and the ability of it to still replicate means that the subsequent progeny could in fact reincorporate that E protein back into its structure and go on to spread infection. Um, and the pathogenicity means that, okay, so the ones that lack the E protein on cell, they can't leave that infected cell, and they die within the cell through apoptosis or other cell signaling, so it dies off. But in other portions, as we just discussed, of the host that's infecting tissue cells, it could very much still be present. So you get a test that tests negative for the E protein but positive for the RDRP. That's essentially doesn't mean you are without infection still. So what's been now determined is that, okay, if you test for one of them, the RDRP is positive but the E protein is negative, it's a inconclusive test, and that patient should still be considered as infected, positive, and all the precautions and isolation protocols should be put in place. And as we continue to progress with this pandemic and this outbreak crisis, more and more tests are being created, and there's a global collaboration of resources that I'm also going to discuss in the testing modality. But I have received an article from Fuzhou China Hospital, and they share with us their testing modality and exactly what proteins they use as their PCR parameters, and also how they go about collecting specimens, not from just one nasopharyngeal swab, but from multiple specimen sites. And again, we're going to dive deeper into that in the next video that's going to come up. But for now, understand the fact that the E protein, very important for pathogenicity and testing. And finally, here we have it. Here is our little guy, our little COVID-19. And I'm going to go ahead and circle as I talk about this up here. We see all these riddled little projections. That's the S spike protein. And we're going to talk a lot about that in the slides to come for the next video. And down here we got our two little orange blobs, the M protein, and here's the E protein, tiny little yellow blob. And over here, this could very well be the HE, fifth structural protein, though I've had a hard time finding evidence to that. But if you can see its close relationship to the S spike on some of these, the close relationship for the S spike and the HE protein, or this white blob, it could stand to reason that it very well could be the HE or hemagglutinin esterase protein. Whether or not it is, I cannot say for certain. But for now, I'll leave you with this image, and I hope that you've maybe learned a little bit more from this first series, and I look forward to talking more about this in the next series on replication. Thank you.